While living aerobically also, E. coli can respond to the changing levels of oxygen. So when oxygen levels are high, E. coli uses cytochrome O, which has low affinity for oxygen. And when oxygen levels are reduced, E. coli uses cytochrome D. This cytochrome D has high affinity for oxygen, so even if the oxygen levels are low, E. coli can carry out its electron transport chain. It can trap that oxygen and use it as final electron acceptor. Now we will come to the systems, regulatory systems that govern the gene expression accompanying the shift to anaerobiosis. So there are basically four systems for regulation of gene expression by oxygen or nitrate in E. coli. The first system is the ARC system. ARC system. The second system is FNR system. FNR. The third system is NAR system, and the fourth system is FHLA regulon. So the ARC system stands for aerobic respiratory control or anoxic redox control system, ARC. This is a two component system. It means that it has a histidine kinase and a response regulator. This system represses many of the genes which are involved in the aerobic condition or which are active under aerobic condition, and it activates many genes which are expressed or which are active under anaerobic conditions or microaerophilic conditions. Okay, so this system acts as a repressor. It acts as a repressor for the enzymes which are active during aerobic condition, and it acts as an inducer or stimulator or activator for the enzymes which are active under anaerobic or microaerophilic condition. The second system is the FNR system. FNR stands for fumarate nitrate reductase. Okay, fumarate nitrate reductase, FNR system, and this is not a two component system. Remember, it is not a two component system. This system stimulates the transcription of many genes that are required for the fermentation and anaerobic respiration. Okay, so it stimulates many genes that are required for fermentation and anaerobic respiration, and at the same time it represses the transcription of some genes that are functional during aerobic growth. In E. coli only, it has been found that there are more than 75 genes regulated by the FNR system. And this FNR system is active only during anaerobic conditions. FNR system is active only during anaerobic condition. The third system is the NAR system, NAR. And there are different types of NAR system designated as NAR L, NAR P, NAR X and NAR Q. L, P, X, Q. This is a two component system and it stimulates the transcription of nitrate reductase and other genes required for the nitrate and nitrite metabolism. This system also represses the transcription of other terminal reductase, for example fumarate reductase or the T-mano reductase, DMSO reductase, etc. This system is active only under anaerobic growth condition in the presence of nitrate or nitrite. Okay. The fourth system is the FHLA regulon. FHLA regulon, and this is also called as formate regulon. Formate regulon. This system consists of genes repressed by oxygen and nitrate and induced by formate. Many a times it happens that the cell do not have the nitrate as the final electron acceptor, neither it has oxygen, but the formate is present. So the formate represses the genes which are active in the presence of oxygen or which may be required for the nitrate metabolism. So those genes are repressed by this formate. The regulon includes several genes including those for formate hydrogen lyase, which converts the formate into the hydrogen and CO2. In FNR system, the regulation is due to protein called FNR which is encoded by the gene FNR. This FNR acts as the positive regulator of the transcription of many genes that are expressed only during anaerobic growth, and it acts as the repressor for certain genes that are expressed only during aerobic growth. You can get a clear idea from this particular picture. So here you can see that the FNR exists in two states, inactive states and the active state. In the presence of oxygen, the FNR is inactive, and when oxygen is absent, the FNR becomes active. So this active FNR shows two types of regulation, positive regulation and the negative regulation. So negative regulation means what? It acts as repressor. So under negative regulation, the FNR acts as the repressor of cytochrome O oxidase, FNR autoregulation and the NADH dehydrogenase 2. Whereas when it is active, it acts as the activator for the different enzymes which are involved during anaerobic metabolism, such as the asparginase 2, then aspartase, DMSO and TMNO reductase, formate dehydrogenase, fumarate reductase, nitrate reductase and pyruvate formate lyase. So all the enzymes which are activated by FNR are essential for the survival of cell under anaerobic condition, that is in the absence of oxygen. Asparginase 2, aspartase, DMSO reductase, TMNO reductase, formate dehydrogenase, fumarate reductase, nitrate reductase and the pyruvate formate lyase. Whereas the enzymes which are repressed by the FNR system or enzymes which are inactivated by the FNR system are cytochrome O oxidase. Remember that cytochrome O oxidase is involved in transferring the electron to the oxygen. Then FNR regulation, FNR autoregulation and the NADH dehydrogenase 2. So some enzymes which are essential under aerobic conditions are inactivated by FNR system, and other enzymes which are essential during the anaerobic condition are activated by the enzyme. Now the active FNR is a homodimer of an iron sulfur protein with an oxygen labile 4Fe4S cluster in each monomer. So it is a homodimer. Homodimer means what? It is made up of two subunits, and those two subunits are similar. Besides that, they also have a cofactor, that is iron sulfur, and this iron sulfur center is oxygen labile, means it will undergo the change in the presence of oxygen. So when it is exposed to the oxygen, when oxygen is available, this iron sulfur center, that is 4Fe4S center, it gets oxidized. 4Fe4S center gets oxidized and it can even be lost from the protein. It can even lost from the protein. And when this happens, a fraction of the FNR loses its iron cluster and it becomes an incomplete protein, that is also known as apoprotein. Both the protein bearing an oxidized cluster and the apoprotein are inactive. Means even if the iron sulfur center is present but it is in oxidized state, or if the iron sulfur center is completely lost, then also the protein is considered as inactive protein. So these inactive protein bind with low affinity to the DNA and they do not stimulate the transcription. They do not stimulate transcription.